Well, hi, everyone. Thanks for being here in the evening. We are recording this for people who couldn't be here, and we'll post it on the Vernal Pool Phenology Project YouTube channel and share that link. So, but thank you for being here. It's nice to have over 100 people here on the kickoff call. So this is the UNH 2024 kickoff meeting for the Vernal Pool Phenology Project. And we're going to tell you a little bit about what the Vernal Pool Phenology Project is and what ERIN, E-R-E-N is, which the Vernal Pool Phenology Project is a part of the ERIN organization, or was created under ERIN. And we're going to tell you about the project, our goals, a little bit about sampling protocols, and where all of us at UNH kind of fit into the whole. And then we'll have time for questions at the end, but please also feel free to ask questions as we go. We don't mind being interrupted, and we also, of course, don't mind getting questions in the chat. I will just say, if you're not intending to ask a question, just make sure you're muted, because we're just hearing a little bit of background noise. But yeah, please feel free to ask questions as we go. Okay.
And it can be out loud or in the chat, because Jen had promised earlier to monitor that.
Did I? I just promised right now that I would monitor the chat. Yeah, Kelsey and I will monitor the chat.
Hey, wonderful.
Cool. So yeah, I guess we should introduce ourselves for those of you, because some of you know some of us and not others, but I'm Jennifer Pernhage, and I'm at the University of New Hampshire. I'm in the Department of Natural Resources and the Environment.
I'm Dave Steinberg, also at the University of New Hampshire, but in the Department of Biological Sciences.
And we have, so Dave and I are two of the co-leaders of the Vernal Pool Phenology Project. And we have two other co-leaders, Mary Beth at Siena College and Thelina at Bridgewater State. And they're not on the call tonight because this is really a UNH kickoff, although we did invite folks from beyond UNH. So hopefully we have some beyond UNH people here as well too. So as we said before, the Vernal Pool Phenology Project was created. We created it under this kind of umbrella of ERIN, the Ecological Researchers Education Network. And ERIN is an organization, a network of faculty that are largely teaching intensive faculty. So people who have really high teaching loads, but and do a lot of their research with undergraduate students, either incorporated into classes that they're teaching or with independent studies and kind of one-on-one mentorship. And the idea of ERIN was to bring all of these teaching intensive faculty together from across the US and beyond, but mostly in the US, to have us kind of leverage the power of this big network of people to be able to use the same field protocols to collect data across large geographic areas rather than doing these really intensive studies locally. And so we're able to collaborate with people all over the place, collect a lot of data using the same protocols, and have some really high quality publishable data from these projects. So you can go to erinweb.org and see any of the projects that exist at ERIN. There are a ton of different ecology projects there. But a big component of all ERIN projects is incorporating undergraduate students in the collection of data. So next slide, thank you. So here at UNH, Dave and I, along with Mike Simmons, who couldn't be here tonight, are kind of the site coordinators, I guess, of the Vernal Pool Phenology Project. So the three of us have been incorporating the Vernal Pool Phenology Project into various classes and working with independent study students and capstone students and honors students working on theses for a few years now. And so we have a list here of some of the classes, I think most of the classes and ways that people are involved in the spring semester, with this project. So we have a bunch of different classes at all different levels that are going to be collaborating together on this project. And and I don't have the map yet, but you will see a little bit later, I'll just say that while we have, I don't even know what, almost 200 maybe students who probably will be involved in the collection of data here at UNH at our vernal pool sites, there are also well over 30 other institutions across North America that are participating in this project along with us this year. And we'll show you a map of those sites later.
Great. So you've heard from Jen a lot about the vernal pool phenology project. So we figured it might be a good idea to tell you exactly what that is. And the best way to do so might be just to put out there the main question that we're trying to answer with this project, with this network of collaborators across a large part of the United States. And that question is, how do both local and regional environmental factors affect the spring phenology of pool breeding amphibians and of trees that are around vernal pools? And in particular, we're seeing whether or not there ends up being any sort of mismatch in phenology of amphibians and trees. And we'll talk about why that might be the case and what that might mean a few slides from now. For part of this project at UNH, regardless of what class you're in, you're probably going to be reading a more detailed description of the vernal pool phenology project. That's what you're seeing up here on this slide. So we're not going to go over that right now. I'm not going to read it to you, but just know that all of this is available up on the website if you're particularly interested tonight, can't sleep, whatever it is. You can head over there and check that out. Now, we've been using the term phenology quite a bit, and you may also be thinking, well, what the heck is phenology? It's really just the study of seasonal or cyclic natural phenomena, and particularly the timing, studying the timing of natural phenomena. So one example might be studying when leaves begin to put their flowers out, right? The timing of flowering in spring or the fall or whenever it might be. Another thing that you might study if you're interested in phenology is when leaf buds begin to burst. All of those specific events are referred to as phenophases, these instances of some sort of phenological trait or characteristic. We call them phenophases. In fact, bud burst, leaf bud burst, and flowering are two of the traits that we are going to be keeping our eyes out for when we're working here at UNH and beyond for the Vernal Pool Phenology Project. An example of some phenophases in animals, if we consider mole salamanders, might be the timing of migration, mating, the arrival at vernal pools where these salamanders often are congregating to mate, egg deposition, when are eggs laid down in the water, also might be another important phenophase to consider. And then you can think about the larval development and metamorphosis as well. When do metamorphs emerge from a vernal pool? Those might be stages that you might be interested in exploring if you're particularly focused on phenology. Now, why might the timing of seasonal activities change? Why are we looking at phenology year after year after year at these vernal pools and across a broad geographic range? Well, you guessed it, it's climate change, right? We all know that greenhouse gases, in particular atmospheric carbon dioxide concentrations, have been increasing. That's what you're seeing on this top figure right here. From the mid 2000s up until 2022, we're seeing a consistent increase in the amount of carbon dioxide in the atmosphere, despite the fact that we all know it's a big problem, right? And sort of joined to this, we've been seeing over the last 150, 175 years, an increase in annual global temperatures. And that's what the bottom figure is showing here. We have time from 1880 to 2020 on the x-axis, and we have temperature anomaly in degrees Celsius on the y-axis. And you can see that where we sit today is quite a bit above what the expected annual global temperature would be. So why does that matter for phenology? Well, how might an animal determine when to migrate from one place to another? Or how might an organism determine when to lay an egg? Or how might a tree determine when to bud burst or flower and all of that? It may have something to do with temperature, for example, or daylight, or who the heck knows. But certainly temperature and some other factors are going to be changing along with or due to global climate change, and we might expect that to have an effect on phenology. In fact, that has been demonstrated a number of times, and in both plants and animals. So this is a pretty well-known study here on the left that's looking at the timing of flowering. And you see that on the y-axis here. We've got 19 May, 19 April. And how that timing of flowering is related to the mean spring temperature in the area that this study was conducted in, which happens to be in the great state of Massachusetts. And what you might notice is that the mean first flowering date for a number of different plants has actually been creeping further and further and further up the calendar. In fact, we see plants that are flowering almost a month earlier in some places just within the last 100 years. This study, by the way, is really cool because the initial data were collected by Thoreau. More recent data by obviously not Thoreau, since he's long been dead. Over here on the right, we see an illustration of the fact that animals also have been shifting their phenology. And in particular, I'm showing you a few different species of salamander up here. If you take a look at the x-axis here, we've got year from 1978 up until 2008, and the trends that you're seeing on this graph have continued for the last 15 years as well. And the median arrival date in days is on the y-axis. If we pick just one of these species here, let's say the red one, right, our tiger salamander, you can see that the trend is that our tiger salamanders have been migrating earlier and earlier. Right? And if we take a look at, let's say, the Ambystoma opacum here, we can see that they're actually migrating later and later in the year. Right? So we have a shifting in the timing of these really important reproductive events, really important life events for both our animals and in plants.
So we in our vernal pool phenology project are going to be monitoring wood frogs and spotted salamanders, as well as trees surrounding the forested pools that we're studying. And we wanted to just mention why, given the context that Dave just gave you about phenology and shifts in phenology related to climate change, why we've selected the species that we have to focus on. So why wood frogs and why spotted salamanders? Wood frogs and spotted salamanders are two of the earliest breeders. One frog, one salamander, but two of the earliest amphibian breeders in the spring. And so we would expect, as Dave was showing us with the mole salamanders, that spring breeding species, early spring breeding species might be where we would see the biggest shifts, the earlier kind of shifts in phenology, and then fall breeders shifting later as the kind of seasonality is changing in response to climate change. And so that's one reason. These are early breeders. Another reason is that they are pretty common species. These aren't known as rare or declining species right now. So pretty common species with wide distributions, which will allow us to get a really nice data set across a pretty good geographic range. And right now, our sites, our vernal pool phenology project sites, extend from, I believe, North Carolina is our furthest southeast site. Dave, correct me if I'm wrong. To Minnesota right now, I believe, is our furthest west and north site. And so pretty nice geographic range in the country. And we keep holding out that we'll get some Alaska folks to join us for the wood frogs. But so those are some of the reasons why we chose these species of amphibians. Also, having a frog that calls allows us to monitor species in a couple of ways. So we're going to be talking about how we're monitoring them via egg deposition, but also having a frog that calls allows us to use acoustic monitoring to try to get a sense of the earliest activity that's happening for wood frogs in the system. So we'll get to that in a moment. And then in terms of plants and trees in particular, that varies quite a bit in terms of which species we're studying from site to site. And so the way that we've done this is we're choosing selecting up to 5 species of woody plants of trees that are growing within about a 5-meter, usually 5-meter, up to 10-meter buffer around each of the study pools. And so which trees are dominant, are the most abundant, is going to vary from site to site. And you'll here at UNH, you'll even see that there's some variation between the 2 vernal pools that we're studying. We have some overlap in some of those tree species, and then some kind of turnover between the 2 sites, some difference between the 2 sites. And so we're using these local species that are very closely associated with the pools to try to see, so that we can potentially see if there's any connection or any potential there might be implications of asynchronies or mismatches that start to occur in phenology between plants and amphibians. If the plants start to change their phenology and that has some impact on the hydrology of the vernal pools, that could actually be affecting amphibians down the road. So that's kind of one of our longer term goals, but that's why at first glance, it might seem kind of bizarre that we're looking at amphibians and trees, but there are some real interesting questions down the road and implications down the road related to that. All right, next slide. So let's get into what we're going to be doing. These pictures, because this is a UNH kickoff call, are all from UNH sites. And I think we have all 3 of our original sites here. Or study pools here. We're now only following 2 of them. Isn't that right, Dave? Isn't the top one that one that we threw out?
It is. That is
Yeah.
the original VP01.
Yeah, which no longer exists because we've never found an amphibian there. Even though it looks perfect, we can't find anyone breeding there. But the pool below it, and I can't point to it, but the pool right below it, well, sure, that one. That one is a fire pond right in East Foss Farm. And the picture to the right of that is another angle of that pond. And then the one that Dave was just looking at in the middle is our second vernal pool. So we have 2 pools that we're going to be monitoring, and we're going to be monitoring at these sites some abiotic measurements. So we're going to be looking at temperature, air temperature, and water temperature. We're going to be looking at hydro periods. So how long do each of these vernal pools hold water throughout the year? When does the pool dry up? This is obviously very important for the success, for breeding success in our amphibians. Usually much more of a problem for spotted salamanders than it is for wood frogs. We're going to be monitoring water level throughout the season. So not just waiting for that moment when there's no more water left, but how do water levels fluctuate and how quickly does the water level go down? And I don't think we've started doing soil monitoring, but we've thought about adding some soil monitoring. And every year we take 2 sets of canopy photos. So when there are no leaves on the trees, and then at full leaf out to try to get a sense for the range of canopy cover at each of the sites. As I mentioned before, for our wood frogs, we do acoustic monitoring, and we've shifted from using the Swift, the big honkin' Swift units that Dave was circling there, to these really nice, sleek audio moth units that you'll see when we go out in the field. And the biggest data point we're trying to get with acoustic monitoring is when is the earliest kind of the onset of wood frog calling at our vernal pool sites. But we also may look at, kind of peak calling or peak chorusing and some other acoustic kind of measures. But the first thing we're looking at is that phonology, that onset of calling of wood frogs at a site. All right, next. And so in addition to the acoustic monitoring, we're also going to be doing egg mass surveys for both species. So for wood frogs and for spotted salamanders. And we're going to be doing these egg mass surveys by doing perimeter searches, so walking around the edge of the pools, not by walking through the entire pool. And because we're trying to limit the amount of disturbance that we're having on these sites. And so we're going to be looking for the first eggs to be deposited, but we do continue monitoring even once we see the first egg mass show up. But we're not doing full counts, but we do kind of notice, oh, there were waves of eggs. So we saw like one egg mass, and then maybe a week later, there were all of a sudden a ton more eggs because there might be like an early individual, a couple of individuals that show up early, and then like a big wave that comes later. So we're trying to get some of that in our data as well. And then it also says here spermatophore surveys. So spermatophores are, Dave's circling a little spermatophore right there. They're essentially just these little packets of sperm that male salamanders deposit on the leaf litter at the bottom of the pools. And the females, they kind of try to entice the females and do a little water dancing in front of the females and get them to come down and pick up those spermatophore packets with their cloaca, and then there's fertilization at that point. So they don't, that's the way that the mating is happening with spotted salamanders. And so seeing those spermatophores can be an indication, another indication that breeding has started. And that can help us out either as an early warning that like the early males showed up and started to put down spermatophores, or that we should look a little closer and try to see if there are spotted salamander egg masses, which can sometimes be in the deeper parts of the pool and a little bit harder to see. So, all right. Dave, do you want to pick up the next?
Just to add one thing here.
Yeah.
I know Jen's been talking about acoustic monitoring, and we do put out these audio recorders that are constantly essentially listening for the calling of frogs, but that doesn't mean that you won't get to hear frogs calling when we're out as well. So when we're out doing our surveys, always make sure that you're sort of keeping one ear open because we have heard wood frogs out there, and it's always exciting to hear that. Additionally, a number of the courses all of the courses will be going out at different times of spring. So some folks will be out there really early on, and you're going to have that amazing excitement of, oh my gosh, there is a wood frog egg mass here. It's the first one of the year. This is awesome. You're jumping up and down. Everybody's taking a million pictures of that first egg mass, right? There's going to be another group that finds the first spotted salamander egg mass. Same thing is going to happen. Some of those later groups, you might be like, oh man, well, there've already been, first off, you get to see the egg masses for the first time that year. So that's always exciting. But also there are other things going on that we always get to observe. One year we saw some leeches slurping down some wood frog eggs at the end of spring. We might already start to see some wood frog tadpoles potentially, or at least seeing some an increase in the development of the wood frog embryos. And that's really cool to see as well. So regardless of what stage you're at, you're going to get to see different cool stuff or experiencing or experience interesting natural phenomena. And I don't want to leave plants out of this. I know Mike
Wait, hold
Simmons.
on before we get on plants, because just one more thing about acoustics. So we had said that the main thing that we're looking at is that first call. That's, what we're going for here is when is that first call happening? But when Dave was mentioning that we might hear frogs calling while we're out there, that is another thing that we're doing. We're taking a lot of audio files right now. And one thing that we're also trying to figure out is, are there times of day or night that are kind of the most where wood frogs are calling the most frequently, or where we're most likely to kind of capture them? Because right now we're just is it every hour on the hour for 5 minutes, or
Mhm.
every half hour? Yeah. So we're collecting data every hour on the hour for 5 minutes, which is a lot of audio data. But wood frogs call day, night, whenever, and we don't want to miss it. And so right now we just have to collect all of these tons of data. And so one thing we're hoping to be able to provide is also just a little bit more, at least in this area, of an understanding of when are frogs calling, when are these frogs calling. So, but now, okay, we'll try to get off of the amphibians now, talk about plants. Go ahead, sorry.
Well, I was just going to say, I don't want to leave plants out of this because a large portion of the project is, in fact, trying to determine when our different individual trees are beginning to flower, when their leaf buds are bursting, when their leaves are fully emerging. And for some of you animal-focused folks, that might seem a little boring. Guess what? It's not. When we get out there, everyone gets super excited to put the binoculars up to their eyes and stare up in the canopy. I'm not even joking. And just like trying to see, is there a little bud that is bursting? Do I see a little bit of green coming out? Same thing. It's exciting to see sort of our community members begin to wake up from a long winter. And that's what we're doing out there, right? Is looking at individual trees and seeing them sort of get primed for the spring and summer. So that also is something that I at least have really enjoyed out there, and a number of students have said they've been surprised at how much they've enjoyed the tree component of this project. We are going to be also including some remotely gathered data into this project. So looking at sort of broader scale, forest types, vegetation types. And down here, you'll see that there are other optional data that folks could collect at their vernal pools. For example, one could set up camera traps and explore the use of vernal pools by other organisms. And in fact, we do that at UNH. So everyone will be involved to some degree, at least, in seeing what other organisms might be using our pools other than our trees and our two amphibian species. And you can see actually one of the guests of our vernal pool down here in this image. Hopefully you can see that.
It
So
might be
wild.
clearer on the next slide,
Yeah,
a little bit clearer.
there we go.
Um, Yeah, so we are going to, I don't think we said yet that the surveys for this project go on for 6 weeks. So we do 6 weeks of surveys. We try to start surveys about a week before we expect, at least a week before we expect to actually see eggs showing up, so that we don't miss it. We already set the audio recorders out, what, a couple weeks ago, a week and a half, 2 weeks ago? I don't even remember. But back when we started getting freaked out because it was really warm and rainy, we went out and set out the audio mugs so that we can hopefully see that. And then everyone in the Vernal Pool Phenology Project at all the different sites is going out for 6 weeks from whenever they begin, twice a week, and doing surveys. And so we try to separate the surveys by at least a couple few days, and do 2 a week. And every time we go out, we are going to read our water depth gauge. We're going to walk the perimeter and look for egg masses and spermatophores, even if we already know that they're there. We just take another look and see how they're developing, or if there are more eggs or eggs we didn't see before, or different species that we didn't see before. And we go out and we visit every single one of our trees that we have flagged. I don't know if we even mentioned that. We'll probably mention it. I'm sure we'll mention it a lot when we go out there, how it's set up. But we have something like about 20 different individual trees at each site that we're monitoring over time. And so we go back to the same trees every year and look at all those phenophases that Dave was talking about. We're going to start our surveys during spring break. Dave and Mike and I are going to go out and do the first surveys while all of you are on a beach or something. And we're going to do the first surveys. And then some of you who are on this call are going to be there for the first surveys right after spring break, starting on Monday after break. And we're going to go out, and that is when we're going to start actually putting up some of our trail cameras as well, and doing some of these additional types of monitoring that we do at the sites. So in weeks 1 and 2 following spring break, we're going to put up one camera per site during that first week, and then another camera per site during the second week, to just try to monitor wildlife that are using the pools. And these are just some examples of some of the species that we've seen. We've had, owls and bobcats and beavers and bears and all sorts of birds. And is there something else that I'm forgetting that was really cool? I bet there was.
Oh, I mean, this
People.
past year, oh yeah, there are people.
Be careful what
Fortunately,
you do in College Woods and East Foss Farm. There's cameras everywhere.
there are a lot of porcupines,
Yeah.
some possums, obviously squirrels and chipmunks and mice. There's this one mouse at one of the sites that it's like every night comes down, like lives up in a tree, comes down the tree, runs all around, goes back up the tree. It's pretty fun to watch. But yeah, hawks, owls, there's a weasel or two in our most recent batch of images. So a lot of cool stuff out there. And if you, and you will be determining where these cameras go, everybody, not us, you will be determining it as students. If you sort of angle things, maybe we see more stuff actually interacting with the pool itself, because we've tended to kind of have these pointed at the perimeter of the pool, but maybe we could even have one camera pointed at the pool and we could see hawks eating frogs or who the heck knows what. So always fun thing to look at.
Yeah. And yeah, we just wanted to mention also that we do have all of these camera data, all these camera pictures from our ponds, and we'll be collecting more. We have all these acoustic files. There's certain information we need for this project, but there's a lot more that can be kind of mined from all of those audio files and those the photos. And so if folks want to get involved, if you're going to be here in the future and you want to get involved in the project or kind of a side project related to it, there's a lot that can be done. And we've already had a handful of students who have done kind of spin-off projects related to the vernal pool phenology project here. Some who have submitted a herp note because they observed something that hadn't been observed before in one of our vernal pools, which we should have put the video in this
But we'll show it during our courses.
Yeah, we'll show it during our courses, but it's a pretty cool video of these midge larvae eating wood frogs that hadn't been seen before. So we are happy to get more and more people involved, and we've had, I think, at least 2 or 3 folks present at the undergraduate research conference about vernal pool phenology project related things. So please approach us if you're interested in that. Anything else you want to say about this, Jean-Paul?
No.
Okay. All right. And then just for any folks who are here, not from UNH, the vernal pool phenology project and like who's participating in it looks different at different places. Here at UNH, we have hundreds of students that we get involved every spring working on this. At other institutions, it's a couple of people doing independent studies that are monitoring one pool. It looks different in all places. Our vernal pools all look different. How much we learn about our vernal pools is different. But we really value the participation of everyone in this project. And it's really fun. I know that Dave was saying before, that some of you are in a couple of the classes that are participating in this. So I think we have some folks here who are in ecology right now and also in conservation biology, for instance. Some folks may be in conservation biology and forest management with Mike. If you're involved in multiple classes, we hope you see that as an opportunity to be to kind of be there throughout the season and to see some of the change that's happening at these sites, and not to just see it as like, oh, I'm doing this for multiple, I have to do the same thing for multiple classes, but instead you get to maybe be there early on for one of your classes, and see, maybe not see any egg masses and just be walking around thinking, how could you ever find egg masses in this murky water? And then you get to be there 2 weeks later and you actually see your first egg mass, and it's kind of exciting. So, and same thing with the plants. As Dave was saying, we're always shocked at how excited we get and how much we kind of geek out about learning about the plant phenophases during this project. So if you get to be there, throughout the 6 weeks because of different classes, or because you just decide to tag along and join some of our later surveys, it can be really exciting to see what happens throughout the season. So, all right. And then
On this slide, I
yeah.
just wanted to
Oh, yeah,
also
yeah.
that first off, even if you are in multiple courses that are working on this project this semester, you may be doing slightly different things every time you go out, right? Like we're always doing these surveys of our trees and for eggs and stuff, but then there are other little different components that are sprinkled in for our different courses and that are sort of tailored to them. Also, year to year, there might be different elements of the projects that courses are involved in. So in this image up in the right here, this was a year when students in courses were actually setting out those water depth gauges. You can sort of see this white PVC pipe.
You can see
Right
on the
here.
picture on the left, someone holding one out of the water. Yeah.
And this year, one of the classes will be replacing some of their temperature loggers and other things. Here over on the left, you can also see students with those water gauges setting those out. So there's always sort of some little different element that you could be involved with.
And I think, oh, this is just, we already mentioned just different ways that folks participate, but this is our map that shows our sites from 2023. So these are all the places that participated. And I think there was one up by Minnesota as well. And then we have many more folks who are planning to join for the 2024 season. I think we have like over almost like 90 people on our email list now. And so, there are a ton of people involved in this project. So you're participating for one class or a couple of classes, or, however you've gotten involved, but you're part of something like really big, a lot bigger than us. And the data that we're collecting, are intended to be analyzed, across this whole region, and published to share information about how phenological change is happening and whether these phenological asynchronies or mismatches are occurring. And so we really value your contributions to this project. And it's not just, kind of a cookie cutter lab that's created, for a class where we're having you go out and learn a technique. Like, you are contributing data to a project that has faculty and students and just community members involved from across North America. So, yeah.
Yeah. And I guess it's good to also say this is the third year that we are doing sort of a full scale run of this project. And so this is also something that people have been involved with for a few years and will be continued to be involved with for many years into the future. So you're part of, a big group of folks who are all working and striving for the same thing right now, but also an even broader or larger group of people if you look across time, which I don't know, that always makes me feel good to think about.
All right. So I don't see any questions in the chat yet. Does anyone have any questions about the project? There's a lot, I'm sure, that we skipped. that we'll talk about during our respective labs and classes. But we just really wanted to give an overview and also kind of, get 100+ people together who are all going to be working on the same thing, as a little mini like pep rally of sorts, to kick off the season. Kelsey, no pressure, but if you want to jump in at any point, feel free if you have any thoughts from participating for a couple of years.
I don't have any questions, but I'm excited that spring is here and excited to go get outside with lots of you. So,
Yep.
Yeah.
Has anybody out there seen any amphibians yet this year, or has anybody heard any wood frogs or spring peepers calling?
I saw a frog over the weekend.
You did? Do you know what kind it was?
I don't know. It was kind of brown though.
It may have been a wood frog, maybe something else. I don't know, but you're gonna be able to distinguish between different species when we're finished with this project this semester, and hopefully get a chance to see another individual of that species out there. So Frankie's saying, are you taking any information from adult salamanders or frogs present at the pool? We certainly note that. And some years, well, actually every year, I think, we see frogs out in the pools, wood frogs and also occasionally green frogs out there. We take note of that, but currently I don't think that's something that we're thinking we're gonna use for some larger data analysis.
Yeah, mostly because we won't necessarily have consistent data across sites for that. But we record it every time we see it at our sites. Yeah.
In terms of adult salamanders, we have not seen any adult salamanders during the day at our sites. I imagine if we went out at night on a rainy night, we'd probably catch them. And you can certainly see them doing that at other places around here as well.
Yeah, Carter, I saw your message about the salamander out at Woodman. Mike shared that photo with us, and we've actually shared another one, right? Of a spotted salamander walking across the snow. So yeah, some have been out and about for sure. And how does participation look for volunteers who aren't currently part of any of the classes? Charlotte and Kaylee, you found out about this, I believe, because you're already on our racer amphibian reptile research list serve, right? And so we will send out, I think, messages to that listserv. Yeah, Dave, to kind of like let people join us if they want to. So absolutely, we'll update you there. And if you're not on that email list and you want to be, just to find out about any of the this project or our terrestrial salamander project or our turtle project, any of the amphibian reptile research at UNH, you can email Dave or I and ask us to be added to that list, and we'll do that. So, and for folks, and Dave's answering it in the chat, but we are going to be sharing a recording via our YouTube channel, and I think we'll all kind of just post that link on our respective Canvas pages, I'm sure. So yeah, and then Nico, let's see, are there paid positions for this? Sadly, no, not right now. Not even for us. But how many spotted salamanders do we typically see on average, and when are the best times to see them? So wood frogs and spotted salamanders are both what are referred to as explosive breeders. Nice. And so meaning that they tend to, much of the breeding population tends to show up at the breeding pond at around the same time. And people always talk about there being like a big night when all of the wood frogs or all of the spotted salamanders show up at a pond. And in reality, most of the long-term studies show that there are a couple of kind of big waves, a couple of big nights, and big waves of animals showing up. But really within like a couple week period, usually a couple few week period, you've got most of that breeding population showing up at the same time and breeding. And that is the best time to see spotted salamanders, especially, is to go out on those rainy nights and try to catch them crossing roads as they're heading to ponds, because otherwise they spend a ton of time either under logs or 6 feet underground in small mammal burrows. And so it's really hard to see them if you're not catching them at that moment. And was there, I feel like there was another question related to that, but maybe not.
Yeah, the wood frogs are a little easier. We tend
Yeah.
to see them out in the pools, at least a couple of them. But same thing, you could also go out on a night when it's raining and above 40 degrees and see them crossing the road. Just be careful when you're out there. There are a couple places on campus. Over by Gregg Hall, there's like this little retention pond that tends to have a lot of wood frog activity. So if you're like really dying to see some of those and can't wait until the vernal pool phenology project lab for your course, you could always head out to Gregg Hall and like a little
to a roadside ditch next to Gregg Hall, which is the
exactly.
best wood frog breeding site on campus.
Yep.
Yeah.
Or back by the Gables. People always talk about hearing them back there as well. That's a little well, I don't know. I've never been to that site. It's probably
Yeah, and
just
didn't you, Kelsey, you were running, I think it was, you were running into them by the equestrian fields, right?
In the equestrian jumps that are kind of just gravel pits that fill up, there are a ton of wood frog eggs in there if you walk past the observatory pond. So
Yeah. The Gregg Hall site is really phenomenal. Like we've gone out there and it's just deafening. The wood frogs calling in the middle of the day, and like you can see a bunch of them in amplexus, these males like grasped onto the females. And I mean, you can basically watch them like depositing eggs. You found like, didn't you, isn't that where you guys found that like ball, that like breeding
yeah.
ball of frogs was
Yeah,
like a bunch of
I
males and females? Yeah.
thought it was a dead frog, but it was a female that had 3 or 4 males holding onto her and holding her underwater. I guess it could have resulted in death. Uh, anyway, how physically exerting will these labs be? Danielle's asking. So the walk over there is the most, I guess, physically exerting aspect of this, and it's about a 20-minute walk from something like Nesmith Hall, James, 15, 20 minutes, something along those lines. And then once we're out there, I mean, the pools are small. We're just walking sort of casually around. There's a lot of standing and looking up at trees and things like that. So the walk, I would say, is the most strenuous.
Yeah, there, and we're only gonna have, we'll probably bring a few pairs of waders out with each group in case someone needs to go in the water to see if that really is an egg mass or something like that. But we're not all gonna be wandering around in the mucky water, especially if you're under 6 foot in the fire pond. Made that mistake.
Yeah, I won't even go back in there.
Yeah. So yeah, it is a lot of just kind of standing around, looking with binoculars, walking around the perimeter of a pond. Not bad at all. Yeah.
One could, it depends on the lab and the course, but if somebody needed to, let's say someone's recovering from, I don't know, knee surgery and they can walk, but they need to keep their walking sort of limited to relatively short distances. There are places you can park over there. So if beforehand, I'm sure you could reach out to one of your instructors and let them know that you would need to take a car out there or get a ride or whatever. Can hopefully make that work for people.
Yeah. Good question. So would wading in the ponds lead to better data? So we've had a lot of conversations about this among the co-leaders and just within the project about whether we should be wading through the pond in order to check for egg masses, I assume. is what you're asking. When people are doing egg mass surveys to do egg mass counts for other research projects, there are some kind of strict protocols that people will follow where you like follow a very like strategic pathway through the water, and you have double observers doing counting. So you have one person start at one end and go one direction and the other person start at the other end. But it can be a little bit destructive. And at some sites, you can't get to the deepest parts anyway, because you just wouldn't be able to wade there. And so we wouldn't be able to get consistent data, we think, across all of the sites in the study. And so we haven't made that part of the protocol. We also don't want to be super disruptive of the site, but we will occasionally, if we have to go in to like, reset a hobo data logger, or if we think we see eggs, but we're not positive that's what we're looking at, because it's kind of like, can be kind of hard, as you'll see, with some of the glare and to see in the water. We might send someone in, but we don't have everybody go in. It would just be really disruptive. So yeah, it's a really good question. We've talked about it a ton, but no, we're not going to all be waiting in there. No. Yeah.
Definitely. And we were also trying to think about how we could have as many people involved in this as possible. And I mean, people in terms of not just here at UNH, but at other institutions and organizations, and thinking about whether folks had access to waders or funds to buy waders, or are people working at remote sites, and really sort of schlepping waders out there isn't really feasible. And so putting all those things together with what Jen was talking about, we settled on this. But yeah, you're right. Like waiting, you're going to get up close to the eggs. But
yeah, and especially if our goal was to understand like abundance, or like how many eggs were there, it would have been a much harder decision. Like we would have had to get in the water. But we're hoping that we can at least have presence, absence, and a day at which we see them, right? That's really what we're going for, is like, are there eggs there, and what day did we see them on? Because we want to know, when the first wood frog eggs are seen and when the first spotted salamander eggs are seen. And so in order to do that, yes, we may still miss some in the deepest parts of a pond, but we feel fairly confident that we can get that without wading in. One of the things that we didn't belabor a ton in this presentation, but that we'll talk about in the individual classes and with handouts that we give to you when you come out to do the lab, is like, how do we distinguish wood frog from spotted salamander egg masses? And other than just, we'll probably see wood frogs first, right? But like, how do they look different? And so that's another thing that not only will you learn the plant phenophases, but you'll also get used to recognizing the difference between the wood frog and the spotted salamander egg masses. Dave, are you going to bring up a picture?
I am.
Cool.
Might as well.
We have some really great images that were published in, I can't remember, was it like northeastern forests or something?
Oh, yeah. Yep.
Some really neat sketches that were done that show like wood frogs versus spotted salamanders versus blue spotted salamanders. And we'll share those with you through the courses. But Dave will maybe tell us the difference here between wood frogs and spotted salamander eggs.
Yeah, on the left here, you can see wood frog egg mass, and you can see it sort of looks like, it's described often as looking like tapioca pudding. You can see each little individual egg, right? All these look like a clump of grapes or eyeballs or boba or whatever. Over here on the right, we have a spotted salamander egg mass, and it's actually coated in this secondary matrix as well. You can see it maybe. It's really, really faint here. And so all of these eggs are embedded in this larger jello-like substance. And it's much firmer. If you picked it up and shook it, it would look funny, but also different than the wood frogs would be. The wood frog egg mass would be more liquidy and would move much more. So there are other ways to distinguish them, and we'll chat about all those when we're out in the field. I'm sure.
And we just had a question about the translucents versus the opaque eggs. And spotted salamanders will vary even in the we're looking at wood frogs on the left and spotted salamanders on the right here. But even within spotted salamanders, you'll see some egg masses that are opaque, that have those white opaque eggs, and some that are more translucent. And I don't know, Dave, do you know if there's a reason? I'm not talking about the algae, but just the opaque versus clear.
Yeah, no, I don't think that anybody has really provided good evidence to support any of the many hypotheses that have been put out there. I think we just don't know.
Yeah, and so yeah, I haven't seen any. So we'll just go with that. We collectively don't know yet. But that does vary a lot, and not just between populations, but within a population. And sometimes there's also a difference between this kind of white and then like what I call like dead egg white. So sometimes you see an egg mass and a lot of them are not opaque like that, and then some are kind of like cloudy and murky, and that can also be a sign that there's something wrong with some of the eggs in the egg mass. But hopefully we'll get a chance to see some of that when we're out there, and you'll get a chance to see it up close and personal. And what was the other thing? Oh, I don't know if we want to mention it really quickly here, if you want to say anything about algae in the spotted salamander eggs, but it's kind of a cool phenomenon that we might get to see.
Yeah, there's this really cool symbiotic relationship between spotted salamander embryos and algae, and potentially mutualistic. It's unclear, but the algae actually grow within the egg mass itself. And it's been shown that the salamander embryos are actually incorporating some of the photosynthetic product of the algae into their growing bodies. So it's not just that the algae is growing on the outside of the egg mass, it's actually in there. And the salamanders seem to be receiving some degree of benefit. You could imagine that the algae, maybe within this gelatinous matrix, are provided some sort of protection as well from grazing herbivores. Yeah,
And maybe
well,
some byproducts for like that they're using in photosynthesis too. Like, I'm thinking about the coral, zooxanthellae relationship. It kind of seems similar in some ways, but
yeah.
Yeah.
Yeah, that's a really good point. And so yeah, well, hopefully we'll see some of that out there. And we can talk more about it. I know we certainly will in ecology. We'll at least touch on it in herpetology as well. So
yeah, you can't stop us from talking about this stuff when we're out there if we see it. So good luck. All right. So it's 7 o'clock. And we want to be respectful of everyone's time because we really appreciate that you kind of relatively last minute agreed to come and have a nighttime lecture of sorts. So thank you very much. And we'll share links to this if anyone wants to refer back to it. We can share the slides as well on Canvas. And we're just really excited to see you out there. Please, if you're here, well, from a class, but also not from a class, if you're here because someone told you we were having this meeting and you joined us and you want to get on our amphibian reptile research listserv for UNH, just email me or Dave or both of us and we'll get you on that email list right away. So thank you guys very much. Have a good evening and we'll see you out there soon.
thank you.
Bye guys. Have a good night.